Hey, Pritesh, are we waiting for you to?
Yes, I was just waiting for everyone to join. It looks like we're around 52 participants, so we'll get started. Good morning, everyone. I'm Pritesh Patel, one of the radiation oncologists here at Winship. I'm really excited today to host Dr. Chan for Winship Grand Rounds. I just want to make an introduction and to let everyone know, if you're an Emory University or healthcare employee and you'd like to receive CME credit for attending today, the login information is going to be in the chat feature on the bottom of your screen. If you're having any issues with the webinar or the CME login, go ahead and let Julie Hawkins know in an email, or you can mention it in the chat. So we're really excited to welcome Dr. Timothy Chan this morning. Dr. Chan is a physician scientist, staff, professor, and attending physician at the Cleveland Clinic and Case Comprehensive Cancer Center. He is chair and founding director of the Center for Immunotherapy and Precision Immuno-Oncology at the Cleveland Clinic, which focuses on immunotherapy research and developmental therapeutics. He is PI of a cancer genetics lab and is an expert in cancer biology and immunogenomics. Dr. Chan is an NCI Outstanding Investigator and an elected member of the American Association of Physicians. He has over 200 publications in journals, including Nature, Science, the New England Journal of Medicine, Cell, and others. He is on the Scientific Advisory Board for Cell and the Frederick National Laboratory, NCI. He received an MD, a PhD in cancer genetics, completed a residency in radiation oncology, and a fellowship at the Johns Hopkins School of Medicine. The Chan Lab specializes in using genomic analysis to characterize the drivers of genetic programs underlying immunotherapy and oncogenesis. These approaches include large-scale analyses, functional genomics, immunogenomics, biochemical and molecular analyses, and mouse modeling. The lab has a special interest in deciphering the mechanisms underlying immunotherapy treatment response, resistance, and mechanisms. So with that, we're really excited to welcome Dr. Chan. Let him get started.
Great, Pritesh. Thank you so much for the invitation. It's really a pleasure and an honor for me to come join Grand Rounds. I guess in this day and age, it's great to be able to at least connect online. Typically, I'm sure the weather, down in Atlanta is way better than here right now, where it's like pretty freezing. So next to the Great Lakes, but maybe in the future we'll be able to connect directly and in person. So like Pritesh said, I'm going to talk about a little bit about our work today dealing with immunotherapy and try to put into context some of our own work, but also discuss areas that I think are important for all of us to know as oncologists about how this field is proceeding. These are my disclosures. So I think that almost everyone involved in clinical or basic research in oncology and cancer research really understand that the tumor microenvironment is an ecosystem that is very rich and very impactful to understand for cancer therapy. You see that there have been major advancements that have led to dramatic increases in patient outcome. And where a lot of the mechanism is based really is at the tumor microenvironment, or TME. And the TME is an ecosystem of tumor cells you see here. Actually, do you see my arrow at all, Pritesh? Maybe I'll use my laser pointer. Let's see.
We can see your arrow.
You can? Okay, well, here, I just switched the laser pointer. So what you basically see here is tumor cells, and you also see a lot of immune cells. And it's this interaction between immune cells and tumor cells and between different types of immune cells in the TME that really dictate what happens. And on the surface, you could imagine that all this interaction in this ecosystem, in this micro ecosystem here within a tumor, is extremely complex and difficult to work out. That's sort of a nihilistic view because you can actually separate out different components and understand the different components and how they function in terms of anti-tumor immunity. This is a way to think about this in our laboratory. We have here tumor cells and T cells here, and you see that there are different aspects of the tumor cell that can dictate immunotherapy sensitivity and response, such as the mutational landscape, transcriptomes, MHC peptidome here. These peptides are a subset of the total number of mutations that are presented. And what this represents is the state of the tumor cell, both transcriptionally and genetically in the DNA, which dictate how these tumor cells can be seen by the immune system as being foreign. And then, of course, you have T cells, a lot of work in the field, including recent Nobel Prizes and so forth, that have allowed us to begin to understand the different states and phenotypes of T cells, which can dictate how active they are in tumor rejection. But I will point out that if you want long-lasting control of tumors, you're going to want immune cells, such as these T cells, recognize tumor immune cells. How this happens really is in this trimeric complex here, where you have antigens that are being presented, right, in the orange, by MHC class 1 and 2 and recognized by the T cell receptor. And this box here really represents the linchpin of what you need in order for the adaptive immune system to reject tumors and to have memory. And so what happens here, right? We all know that the immune checkpoint targets at the immune synapse is very rich in different components. What you see here is very complicated, but I just put this to impress upon you that there are many second and perhaps even third signals that modulate what I circled, the MHC class 1 and 2 peptide, cognate peptide TCR complex. And recent approved targets that have been making a lot of headway into improving patient outcomes include PD-1, PD-L1, CTLA-4, and others, TIGIT most recently with accelerated approval, that are part of these second signals and this regulation that happens to modulate adaptive immunity. There are other targets that have been tested, and by all means right now, I'll just simply say that there have been many, many targets tested in the last half decade after PD-1 and CTLA-4, IDO, ICOS here, OX40, many. None of these are really approved yet because it's unclear how these function, in what context, what tumor types. But we are making headway as a field in identifying different options to target some of these linchpin approved targets. And clearly you can reverse the energy or exhaustion of T cells and let them again recognize antigen-presenting cells and tumor cells and target tumor cells for destruction. So how does this happen? Well, I point out initially this little box here, right, peptide. Well, it turns out a number of years ago, this work actually started about 10 years ago in our lab, we showed that mutation and neoantigen burden predict response to several, response to anti-PD-1 and anti-CTLA-4 in a variety of different cancers. I show here the data from non-small cell lung cancer. This was Keynote 001, okay? So very early on, non-small cell lung cancer patients. We did this in collaboration with Rizvi, my colleague at now Columbia. And what we basically did was treat patients with anti-PD-1. This was second line after initial therapy of chemotherapy. And sequencing the exomes really quickly showed that mutation load, or the total number of mutations, did have a bearing on who responded or not. So here you have durable clinical response, no clinical response. The y-axis here shows total number of non-synonymous mutations, just very quickly just adding up without any other fanfare or details or anything like that. We showed that basically the more mutations you have, the more likely you are able to respond. And Kaplan-Meiers here show a very nice difference. So what this basically implies is a couple things, right? So not just simply that TMB may or may not be a biomarker or what have you, but really the true implication is that the somatic mutations here are, at least in part, part of the important targets which underlies the therapeutic index and the targeting of immune checkpoint blockade, right? So previously it was known that you activate T cells, fine, your T cells are activated, but what in the world is being actually recognized and targeted at the tumor cell level? And this really implies on a large scale that it really is the neoantigens that, at least in part, underlie the specific targeting tumor cells. So what's a neoantigen? So neoantigens, just so that we all know, I'm sure many of you have followed the literature, are normal proteins that in cancer get mutated. And when they get mutated, what happens is potential areas that are presented by MHC class 1 and 2, which happens in all of our cells, at least for MHC class 1, as part of the viral sensing mechanism. And when mutations occur in normal proteins that are usually presented, what happens is they can become seen as foreign and T cells can attack. You can also make neoantigens in a bunch of different ways. This is a little bit of a complex slide, but I'll simply show that, yes, you can mutate the area that the CDR3 from the T cell receptor sees, but you can also mutate the peptide to make something that was previously not presented on MHC seen on MHC. You can generate a new peptide. You can also make frameshift mutations. You can imagine that if you put an addition or a subtraction, you can cause the frame to be completely different, and then you have a long chain of foreign peptides, like bacteria, basically, right? Something that the immune system has not seen. You can also have epigenetic reprogramming that maybe a mutation occurs in the epigenetic regulator, and then you have these tumor-specific antigens that normally aren't seen by the systemic immune system. I will impress upon you that this complex here, this MHC peptide and TCR complex, really has a lot of diversity. Mutational load here, as I showed you before, really is a function of mutational diversity, right? So the more mutations you have, the more potential peptides you can put in. The numbers are calculated for you here. MHC and TCR are also designed by nature to be very diverse, diversity-oriented protection systems that the immune system relies on. And you can generate a lot of diversity. This theme is going to become very important as we move forward. So neoantigen reactivity, this occurs in immunotherapy in immune checkpoint treated patients. This is a patient very early on from Keynote-001 that had his index lesion shrink down. His clinical response actually was excellent, over 90% shrinkage and then stability, doing very well. And what we showed was that we screened a lot of these different mutations that we saw, and we were able to find some mutations that are specifically interesting. They track the T cell reactivity as measured by tetramers, actually tracks with clinical scenario what's happening. So upon treatment, the T cells that react against this specific neoantigen are HER1, it's a passenger event actually, that is recognized by the T cell when loaded onto MHC, really goes up and then subsequently comes down and then stays stable as as a patient reaches a response plateau. There are other neoantigens that we've mapped, and now this has become like a big area of interest in identifying neoantigens. All sorts of papers coming out. These are some of the first in the field that we described. These were patients with melanoma treated with IPI, anti-CTLA-4, and we screened patients and we found some neoantigens that associated with response. You see here 2 of them. What's very interesting is that we wanted to know a little bit about the kinetics of neoantigens, and you see here the top one versus the bottom one. You see very different kinetics. Here you see a gradual increase as IPI is put on board, and then so you see a burst at around week 6. Here you see nothing, and then week 24 you just go boom, like a huge activation here. So it's really quite interesting how not all neoantigens are the same. So there is a pharmacologic diversity in terms of reactivity that develops. And why this occurs is unknown. Maybe partly it's due to tumor kill, maybe the immune system for different types of peptides that are recognized require different amounts of time to really kick in. Not too different than vaccination after viruses, right? Sometimes you get quick engagement of the immune system. Sometimes it takes a booster or more. So very, very interesting. So what about this concept of tumor mutation burden? It's been highly validated now by probably several hundred papers, including prospective phase 3 trials. This is one of them. This is a post hoc analysis of BMS 026, which was a trial of nivolumab, first presented by Solange Peters in 2017, first-line non-small cell lung cancer. And basically, what they showed was that mutational load was predictive of benefit. In high TMB patients, you had nivolumab patients doing better. This was significant. In low and medium TMB patients, you had, if anything, the reverse. And there have been many studies like this, including MYSTIC, a variety of other prospective studies, also including Checkmate 227, as I'll mention. So we asked the question internally, though, whether mutation burden predicts survival for immune checkpoint blockade in multiple cancer types, or was this really only something that was operational in melanoma or non-small cell lung cancer. So we went ahead at Memorial Sloan Kettering and sequenced using a clinical panel that's FDA approved, MSK Impact. We sequenced about 1,600 patients treated with immunotherapy, and also about 9,000 patients that had never seen immunotherapy. And at least in a pan-cancer perspective, you can always see here cutoffs. Just happens to be the optimal cutoff, separating people who do better or worse based on some TMB criteria, whereas you never find this with non-ICI treated patients. This was done by Rob Samstein, who did his Holman pathway work in my lab. He's a radiation oncologist. He now has his own lab at Sinai. We're able to actually break down the tumor mutation load across and its associations with different cancer types across multiple different types of cancers. And what we show here is a forest plot. And if you actually move this way here, this is a decreasing hazard ratio. This is an increasing hazard ratio associated with greater mutations. And these are all the different cancers. And you can see that for many cancers here, what we see is that higher TMB associates with better outcome. And it's very important to note, because since then, I'll mention more about this later, TMB has been approved by the FDA as an indication for treatment with pembrolizumab, pan-cancer. Their cutoff was about 10. I think at best that is a start. I think it does provide an accessible drug to people who often have no other hope. But the cutoff here clearly is dependent and can be optimized. It's not just 10, right? 10 mutations per megabase. And so there's quite a work for the field to do to really optimize the accuracy, positive predictive value, negative predictive value of NGS assays, such as measuring TMB. On the other hand, you can take the approach such as trying to look at how patients with MSI or mismatch repair respond. Luis Diaz, who's a very close colleague of mine at Memorial, when he was at Hopkins, he ran this trial, which is at that time a phase 1. Since then, this has been validated by phase 3s. And what they did was something that was related, right? They took mismatch repair deficient tumors. And as you know, MSI positive tumors have a lot of mutational load, right? Very high mutational load. And so what they found in their phase 1 was that MSI positive patients did very well. This was sequencing the exomes, and what you can see here is mismatch repair deficient tumors had a lot of mutations, and they did actually much better in terms of progression-free survival. And biochemical response here, right, tumor marker level, the blue ones were all the MSI positive patients that did better. And in this waterfall plot, you see the same thing. Keynote 177 was the application of this same concept. You take the MSI positive patients, which have a lot of really high TMB, right? This was done by Merck, and they treated patients with pembrolizumab, and again, the high TMB patients the MSI positive patients did better on pembrolizumab than chemotherapy. This was colon cancer. And the relationship between TMB and response, or the sense that a tumor has a higher amount of non-self, really has been validated over time. Since our initial publications and descriptions of this, there have been a number of different types of tumors different disease types, different histologic types, where this relationship has been shown, and it's still ongoing now. Keynote 158, I'll bring up, is very important. This was the one that was run by Merck that led towards the first approval, pan-cancer for high TMB patients. They took different types of tumors and segregated by TMB high versus low, defined by mutational count per megabase greater than 10, and you can see a better response rate across multiple types of tumors. The numbers are still pretty low. This was accelerated approval. And again, I think that over time, as other drug developers look at correlatives and companion diagnostics, I think that number, that cutoff number, can actually be optimized. So where are we in terms of genomics-based anti-PD-1 FDA approval? Well, for MSI positive or very high TMB, you can have, the FDA has approved for hybrid-mutated tumors that are MSI positive. This is pan-cancer, and this is for patients that have progressed on other therapies. This was approved in 2017. Nivolumab plus ipi-nivo also followed for colon cancer. TMB high was approved as just a simple metric, regardless of MSI positivity. MSI positivity only makes up a small proportion of patients that are TMB high. And there could potentially be other indications, but suffice it to say, the FDA approved pembrolizumab for advanced metastatic solid tumors, progressed on prior therapies. And then there's a first-line appointment for MSI positive tumors. The FDA approved pembrolizumab for colon first line based off of 177 that I showed you before for metastatic and resectable cancer. So this is now standard of care. So it turns out that TMB associates with better survival in ICI-treated patients, but not in patients naive to immunotherapy, right? There's a question of, okay, well, you have better response, but if you have a lot of tumors, tumor mutations, somatic mutations, does this mean that you do worse? Do you do better? Maybe the immune system just fights it off. Maybe this is a prognostic. We just came out with a paper just last month looking at about 8,000 patients or so. And the bottom line basically is that the association between TMB or more mutations and better response and survival is really only seen in patients after ICI administration. If you beforehand and patients without ICI, you actually, if anything, see the opposite, right? So in multivariate models in logistic regression, we are able to actually see this effect take place only at the time after ICI initiation. So you can see here broken down by different disease sites how this shifts over. And so what we basically understand now that we didn't before when we all started this work as a field is that mutational landscapes and immunotherapy are intertwined, right? So you see here the typical Michael Lawrence-Graham here for the TCGA. They basically ranked different mutational burdens across the field. This is a very common slide that people like to show. Mutations can be generated by a lot of different ways, right? So mutational landscapes, smoking, and what have you. And it really is this high mutational load group that responds quite more easily to single agent immunotherapy. So here, along this line and also sort of here. There are a lot of exceptions to this, like kidney cancer that do respond, and where TMB really doesn't track with response. So I have more to say about that. But this really is important in terms of us thinking about how to triage different therapies for new clinical trials, right? It looks like if you have a complex genome and you constantly, make mutations that are different and you have a lot of genetic diversity, then immunotherapies, which really target that diversity, right, which really target that non-self, may be having a stronger role in terms of control those types of tumors. If you have targeted therapies which depend on the presence of a mutation that causes a sensitivity or addiction, you really don't want a complex genome, right? You want something stable on this side, really here, that preserves that sensitivity. Because if you have a lot of, genome evolution, you can actually come up with ways to be resistant. Now, Eden Rubin actually published a meta-analysis of all this, which has been very informative. They've taken a lot of the data, including some of ours, and showed that there's a triple axis of immunotherapy response. You have mutational load here in orange, which really represents this sort of body of variables that represents non-self, right? Intrinsic non-self. And you have T-cell abundance, what is happening in the microenvironment, what type of agents or what type of factors allow things to come in or out of the tumor microenvironment and enable T cells or other immune cells to enter or be excluded. And of course, you have checkpoint targets here, and PD-L1 levels are obviously an FDA-approved indication for different types of cancers for treatment, like lung cancer, not lung cancers, but the level of checkpoints that are being targeted is actually important too. So, how do these comments about neoantigens really, what do they reveal about how immunotherapy shapes different tumor cells during therapy, right? So we wanted actually to answer this question. If the neoantigen hypothesis is true, what we should see is that when you treat patients with immunotherapy, you should see that within tumors, you get a culling, you get a killing of neoantigen-bearing tumor cells. And this is exactly what we see. We actually worked with BMS to run a biomarker trial, 68 patients, in which we took samples pre-treatment from an index site, and then patients received nivolumab, and then exactly 4 weeks later, the patient received a second biopsy that's on treatment at exactly the same site. These are a little bit harder to do now because these are approved agents. On treatment biopsies are really informative. Glad we did this, but this is a challenge to do. And also patients at week 24 had the standard RECIST done using CT scan. And I'm not going to get into the weeds here, but suffice it to say, what you can do is actually compare pre and post tumor genomic landscapes, and you can plot the tumor evolution. So here you have progressive disease, stable disease, a CR, and a PR, and what you can see here, you can track clone by clone. These are allelic fractions that are plotted. This is pre-therapy, this is post-therapy. You can actually draw out different clones that are destroyed and so forth. And when you sum up all of this data across all the patients and put together these scores of genomic contraction or persistence, what you can basically find is that neoantigen-bearing clones, when they contract and are destroyed, correlate very closely with better response and better survival. You can actually plot this out, and it makes a huge difference. And what's interesting is that what's happening here is actually at 4 weeks. This is before any visible change on the CT scan or anything that is visible by imaging, right? So already you get this molecular mechanisms of action that are happening very early and very quickly, just only a few weeks after initiation of immune checkpoint blockade. So again, hopefully what I've shown you is that this part is important, okay? So this part is important because it actually sets up the ability of cells to be recognized as either foreign or not, and that is actually the therapeutic window, the basis of the therapeutic window of a lot of this immune checkpoint blockade, at least in part. I'm not saying that this is the totality of what we're seeing, but this is quite an important aspect of why certain tumors work and how different immune cells like T cells are able to recognize tumor cells. What about MHC? So this is an area that few people tackled. We immediately realized that if you have diversity or mutational load really driving response here, well, the MHC proteins are actually the most diverse set of genes in the human genome. And this has been pushed by evolution for species survival for humans. And this is important because we have a lot of pathogens, and MHC molecules enable us to fight off bacteria and viruses. So they present self-informed peptides to the adaptive immune system. HLA diversity is selected over evolution. We are becoming more and more diverse to fight off new insults to our species. And diversity does provide better resistance against infectious diseases. And this was first posited by Doherty and Zingernagel, right, in their Nobel Prize-winning work, identifying the transplant factors, right, which ended up being HLA. And they posited that the more heterozygous you are, the better your defense is by diversity against pathogens. And this is really proved to be correct. The heterozygous advantage is, or defense by diversity, is evident in populations all across the world that are exposed to different types of diseases, such as HIV, hepatitis B, and so forth, where if you're more diverse at your HLA, you actually do much better. So we asked the same question, okay, well, you go back here and you see, oh, well, this really has to play a role in tumor rejection, because this really is implying what this really is dictating what you can and can't present in terms of somatic mutations. And so we looked, and the first pass of this is that Diego Chao in my lab, who's a fellow in my lab, who's now faculty, actually looked at zygosity. All right, so the same thing that these folks from infectious disease looked at. It basically showed that if you're more heterozygous, you do better. And if you put in a combination score of more heterozygosity at HLAs and more tumor mutation load, you start really separating this out. And actually, HLA is an independent predictor. And it makes sense because, the HLA structure here that actually binds a cognate neoantigen here really is, a source of massive diversity. If you have more diversity here, you can bind more of this and hence make this functional, right? So it kind of makes sense. But there are ways actually to measure HLA diversity that are better than just counting different alleles by their names, right? And that is really molecular diversity. These peptides, these peptide binding receptors have marked biophysical properties that dictate what they can and can't bind. And so we came up with a score called HLA evolutionary diversity, or HED, which actually measures the total diversity of your genotype or patient genotypes. And what we're able to show is that if you have high HED, these patients do better. If you have high TMB, they do better. And if you combine them, you really start separating out the true responders. These are all melanoma patients. This is in all patients, and this is only in fully heterozygous patients. So 6 different alleles on HLA A, B, and C, MOP and POP, or Watson and Crick. So what happens to the immunopeptidome? Well, if you have higher HED, you can actually bind more viral peptides. If you have higher HED, you can bind more self-peptides and also more neopeptides here. And so what is happening, we think, is that the higher your HED is, the better you're able to actually present or make use of your epitope diversity. And it turns out that in a clinical trial that I showed you, 038, we actually did measure the effect of HED on TCR clonality in patients post-treatment. And what you can see here is that the higher your HED is, the higher your evolutionary diversity is, the more clonal your TCR expansion is. So this really does have some functional impact on the T cell activity after immunotherapy. So where are we in our understanding? Well, this is again that complex that I'm showing you, MHC class 1 bound to neoantigen. This is measured by TMB. This is measured by HED. And together, I think they provide better prediction of how tumor cells will see cancer cells. And so we're coming up with some potential thoughts on this and how we can make sense of all this. At least from the genetic perspective, I really start thinking that there has to be genetic tumor rejection potential, right? If you don't have this, you don't have the genetic basis underlying that ligation of the trimeric complex of TCR, MHC, and cognate peptide epitope, then you're not going to get durable engagement of the adaptive immune system. And so the genetic basis of TRP can potentially be a function of ligand diversity as well as receptor diversity in cancer. And right now, just to discuss this a little bit, we're getting very involved in sort of some anthropology genetic studies looking at HLA diversity all across the world. Very, very interesting because this potentially has can provide input on who's responding, who's not responding in different trial patients all across the world. And what's very interesting is that you have something interesting happen. You have something fascinating happen here. All right. So, previously I showed you that HLA diversity here can actually span a large, score, right? So when we actually measure the diversity across the world, across all different types of ethnic groups here, what we really see is that there is almost what looks like a convergent number here, an asymptotic approach to this level of around 6 to 7. Now, why that is unclear. We're actually looking because many of these different subgroups have very different HLA alleles. But why do they all, approach this value, right? Is it possible, just hypothesizing here, that if you're too low, you don't fight off, infection, you don't fight off cancer, like I showed you. Hopefully I convinced you by that. But why is it that, we're not like the Borg, right? Why in Star Trek, right? Why is it that we're not approaching massive diversity, more and more diversity? Why is it that we're sort of hovering around this level, right? So over time, can we actually reach this level, or is it potentially disadvantageous? Maybe you have autoimmunity up here, right? Maybe I showed you here that you're presenting more self-peptides, right? So if you have diversity, HLA, massive molecular diversity here, you can fight off your viral peptides, but maybe you're presenting too many of your self-peptides. So who knows, right? So I think these are all areas that we're looking into with fascination. So moving forward, let me check my time here. Moving forward, we've gone on to really try to map out what other factors influence T-cell immunity to tumors. And in this most recent study, also published, first authored by Rob, who's a radiation oncology resident, we actually used human data here to map out the different pathways that are mutated in patients that were treated with immunotherapy. These are about 2,000 or so patients, 2 to 3,000 patients. And this is a hazard ratio here. This is p-value of significance. And what you can actually show is that a lot of pathways that correspond to better survival and better response to immunotherapy checkpoint really involve DNA repair, right? A lack of DNA repair. Interesting enough, homologous recombination. And pathways that correlate with worse response include things like Wnt pathway, p53 signaling, and so forth, which actually have been studied by other people as well. And so we followed up on some of these genes. Pertaining to this TMB hypothesis, MSI is not the only way you can get high TMB. If you remember back from genetics or oncology, you remember that Pol E, polymerase E, and Pol D1 is a source of hypermutation. If you get mutations in these genes, you can get very high mutation rates. And we're able to show that in these cases, these patients benefit very much from ICV treatment. Here, you have one index patient from a memorial sloan kettering. You can see an index lesion here. This shrinks after treatment with immune checkpoint blockade. We have a small cohort of patients here, and we show that these patients do much better if they're positive for the driver signature for Pol E and Pol D. And we're able to show this in mice as well. We've taken multiple isogenic lines and looked at the effects of Pol E mutations in these models. These are mouse models. And if you have the mutations that are orthologs of the human mutations here, like this hotspot mutation, you can actually see dramatically sensitized to immunotherapy. And even before and then after anti-PD-1 treatment, you can actually see dramatic increase in T cells. This is CD3 staining in tumors that have the Pol E mutations. So this is causative, okay? And again, it's really supporting this idea that the more mutations you have, the more recognition by the immune system can happen. Now, what about BRCA? Right, BRCA is very interesting. You saw up here homologous recombination popping up. BRCA is very interesting here. It's not really clear how BRCA mutations affect immune checkpoint blockade. We wanted to really look at this in depth. Our human data basically shows that if you have a BRCA pathway mutation, you actually do better. Okay, these are memorial sloan kettering treated patients, examined using MSK impact, and by multivariate analysis, we actually showed that it's actually BRCA2 that provides most of the effects. That's really interesting because we didn't really expect this. We thought that BRCAs probably behave the same. They all cause differences in HRD, or they all cause problems with homologous recombination repair deficit and so forth. But very surprising that actually BRCA2 caused a marked sensitivity, and the BRCA1 effect was not very good. And we were able to show this in mice as well. We took syngeneic lines, multiple different lines, multiple different clones, knocked out BRCA2, and what we find is that it does sensitize to immunotherapy treatment. Looking at the tumors, the tumor histology, you see that you get a lot of T cell infiltrate here if you have BRCA2 mutation. For BRCA1, this doesn't happen. In mice, knocking out BRCA1 really does not sensitize to immunotherapy, just like in humans. Why does this occur? Short end is that it's likely that BRCA2 also has a lot of transcriptional repressive activity. In BRCA1 mutated tumors, you have this immunosuppressive environment that happens that doesn't occur in BRCA2 mutations. A lot of immune signatures actually are altered in BRCA2 versus BRCA1 patients, including antigen presentation, T cell activation, and a deep analysis really reveals, using single cell, really revealed that it really boiled down to the myeloid population. Something about BRCA1 allows immunosuppressive myeloid cells to really stay in the microenvironment, whereas this does not occur in BRCA2, and you can actually get T cell engagement because of the lack of that myeloid population. So very interesting. I'm not convinced this is related to TMB, actually. There are other effects, I think, that are in play here. What about in tumors that are that are responsive, where TMB doesn't seem to drive the sensitivity? I mentioned RCC at the beginning of the talk, right? There have been a number of papers now, and incidentally, RCC right now, renal cell carcinoma, is really undergoing a paradigm shift in treatment, right? We've just had really great approvals in the last several weeks with combination of TKI plus immunotherapy that are really quite effective. Now, this is a disease that when I started oncology, right, we're treating with, IL, different interleukins, IL-2 and so forth, and it was terrible. It was toxic. Response rates were in single digits. We're really at a different, happily, we're really at a different phase in the development of treatment for this disease. So this is great for patients. But what you basically see here is that TMB really doesn't drive a lot of this, right? This is TMB, responders, non-responders. Something interesting is going on. This is a low TMB tumor. And so with Ari Hakimi, we've actually looked in depth at what this occurs. This is some other data from McDermott, which we've also used, which shows that actually in atezolizumab anti-PD-L1 treated patients, there's specific signatures that track with better response that really have to do with angiogenesis and myeloid population. So this is a recurrent theme of myeloid population here, right? So we're looking at this really in depth, but this is something that is really interesting. Low TMB patients, high myeloid infiltration. This seems to be, another biology that a main part of the biology of different types of tumors and their ability to respond. So we have to work out as a field. Now, incidentally, when TMB is not operative, it looks like the HLA side of the coin perhaps is. So we ran a trial with Joe Lee, Bob Moser, and so forth, and Renzo here, who's now in residency, and we were actually able to show that patients respond very well to lenvatinib plus pembro. Okay, these are all advanced RCC patients, very good response rate. And the only real biomarker in this case that really segregated response was HLA diversity. You see here PFS separated out. You see here difference in duration of response. This is critical, right? When you have everybody responding by RECIST criteria, at least by the third month, durability is really important. So durability is actually driven, we think, by HLA diversity. And what you basically see here is that this is a largely dose-dependent effect. The higher your HED is, the better your PFS is. And I'm going to basically end soon here. I think therapeutics in the next 10 years is going to be fascinating. Not only do we have an increased understanding of how tumor cells recognize, immune cells recognize tumor cells, and FDA approvals based on this, and indications that potentially can be explored, but also direct therapies that may evolve. And you guys may or may not know, but the current Pfizer and Moderna vaccines, RNA vaccines, prior to the COVID pandemic, these were all platforms geared towards cancer, right? So based on our data and that of others, I've showed you neoantigens or somatic mutations are targeted by cancers, and hence the thinking is that you can make vaccines out of them. And this was a whole cottage industry, right? Moderna and BioNTech were companies designed, that were formulated at first for cancer, right? And so it was repurposed for COVID, and thankfully they worked. But I think that once COVID is under control, you're going to see just an explosion of these types of efforts now, because there's so much more experience with the COVID clinical trials that have been done in science. optimize these type of therapies. Well, in fact, you can actually make neoantigen personalized vaccines that increase the number of T cells that are specific for these specific targets. There have been a whole bunch of trials. I'm not going to go into this, but there's an approach to actually generate individual types of vaccines to target different types of tumors. This has been personalized based on genome sequencing. And this is really a complicated slide, but I just put it up there for reference that you can look at. This is a summation of all the things that we've talked about and some of the things that I have not had time to talk about. But tumor immunity is a very complex phenotype. And as a result, you see that there's been a number of different things that could potentially influence the success of agents that are designed to boost tumor immunity. And so I recommend you guys look into this if you're interested. So in summary, what I'll basically say is that genotype determines phenotype. Immunology follows this rule. We see here that the TCR antigen MHC complex really drives tumor rejection potential. This is something, in fact, is probably pretty expected, given what we all know about the immune landscape and immune mechanisms. Mutational burden is a pan-cancer IO biomarker, one of the first, actually, and along with MSI to measure antigen diversity. There are a lot more that can be done here. The low-hanging fruit really is to identify and potentially look at companion diagnostics for different types of cancers that have mutations that drive high mutation burden, right? So these are pure populations like POLE-E, POLE-D, HRD, and so forth that could potentially be used as to identify patients that respond. HLA diversity is important, very important to really look into this. When you have a vaccine trial, right, you always keep track of your HLA and genotype and diversity. There's no difference with cancer. I think these are essentially in situ vaccinations with a drug, right? And HLA is going to be critical, and we're going to have to look into this. Patient genotype diversity is just as important, I think, as tumor genetic diversity and I think personalization of immunotherapies. So with that, I'll end. These are acknowledgements. Great people that are working on this. I moved to Cleveland Clinic to run this new department about 10 months ago. So we have a new group here. Some of the people have moved over as well, but the folks in red are the really talented and fantastic people that I have had the pleasure to work with. And incidentally, there have been a number of radiation oncology residents. What I basically say is that you'd be a good physician first and study the tumor type, right? Not the modality. And so I during training, I recognized that it's an opportunity to really explore a lot of different things. And so I think that as you're choosing a project or scoping your own career, just think outside the box and focus on something you're passionate about. Right? You can just have a chat with the residents afterwards. Collaborators at MSK and also at Cleveland Clinic, other collaborators and support. Thank you so much.
Well, thank you so much, Dr. Chan. That was a wonderful talk. So everyone, if you have questions, please use the Q&A feature. I think we already have a couple questions there. Dr. Chan, I hope you're able to see that Q&A.
Yes.
Okay, great, yeah.
Well, there's chat and also there's Q&A. Do you want to moderate?
Sure, yeah. Why don't we start with the Q&A questions? Let's see. Looks like Dr. Lazinski has a question. Why do you think the tumor mutational burden diversity only predicts outcome when patients get an immunotherapy regimen versus at baseline, where it does not predict? This is really interesting and perhaps shapes how we think about immunosurveillance theory in human patients.
Yeah, that's a great question. What we're looking at now is the effect of molecular diversity of HLA in different indications and in different clinical scenarios. So one obvious question is, does HLA actually perhaps account for that missing understanding of why certain people develop cancers and certain people don't? So you can imagine that there are a lot of people, well, you don't have to imagine, but let's consider the fact that many patients smoke or many people smoke, And every one of us has, like, knows this guy who's in his 20s that gets like a cancer from heavy smoking very early on. And then you also have this uncle, of course, that's 100 years old, that smokes 50 pack years of history a day and never gets a cancer, right? I mean, there are anecdotes of all over the place like this. And is it luck, right? Is it simply the ability of mutations to occur or not occur in different cancer stem cells? We don't know. But it's clear that the immune system is a sieve, is a filter for any cells that may go awry, that get mutated. So, you know, it's a very reasonable hypothesis that you know, risk of cancer in high-risk patients really is influenced by HLA diversity. Unfortunately, it takes a huge number of patients to actually address these questions, right? Because there's so many confounders, right? Like how much you smoke, you know, other things in the environment, you know, yada yada. So I mean, you know, these types of epidemiologic studies I think are going to be really fruitful. I do think just at the beginning of really understanding and taking advantage of our knowledge of the immune system diversity. This molecular metrics for HLA diversity have only recently been a thing, right? I mean, you know, we were really the first to apply probably, you know, the Grantham distance molecular modeling technique, which has been around since the 70s. You know, I don't know why people haven't looked at this before, but I think that's one thing that's going to be very interesting. The other thing is in terms of different people all across the world. It's clear that there are elements that are outside of HLA in different ethnic groups, and then, you know, other germline alterations, or not alterations, but variations. And then there's HLA. And in a lot of cases, I have data to show you, but there's in certain populations like you know, that don't have as much travel, what's going in admixture, what's going on is that HLAs undergo a whole separate evolutionary process. Okay, so the evolution is driven by pathogen exposure and so forth. In other populations, like in North America, we've done these measurements. It's very clear that admixture is critical. So when you're having a melting pot, literal melting pot, we can detect at the HLA level the extent in which the genomes are mixing. Okay, and it's really, really interesting. And so you have to control for all that when you do these worldwide studies. And so this is out of the realm of something that, you know, a single lab can do. It might be something very important to look at as we do these larger, you know, seer bubble analyses, you know, over different populations. That's a great question, though. We've been thinking a lot about this, and I think there's a lot potentially one can do.
Okay, great. I had seen a couple other questions in the chat earlier. For some reason, I'm not seeing them now. Please feel free to re-enter them, and we'll sort of clear them as we go. Dr. Koo asks a question: have you looked at other factors that impact ICB therapeutic efficacy in mutational load high versus low patients, such as abundance of CD8 cells in TME, immune suppressive cells, PD-L1 expression levels of tumor cells, expression levels of other immune inhibitory or co-activator receptors on T cells, etc.? Is there any way to develop a holistic system predicting potential response to ICB therapy accounting for these factors?
Yes, you know, Dr. Koo, this is a great question. Where the field is headed, I'm convinced, is towards nomograms. And we've been through this with breast cancer, right? I mean, all the oncologists, you know, in the audience know that, you know, back in the day, you start with clinical, you know, staging, and now we have biomarkers such as ER/PR positivity, HER2/neu, and a variety of other types of variables, both molecular and clinical, that we've now put together into nomograms, right? And they're even available at websites called adjuvant.com. This is where we're headed for immunotherapy. It's very clear. And there are a number of, I'll just suffice it to say, there are a number of groups, including ours, that are in the process of putting out models now that take into consideration all these different factors. They need to be weighted properly. So many of us have used multivariate analysis and machine learning to actually weight them to develop models. And that's exactly where we're going. So it's a great question.
Okay. Great, go ahead.
Let me see here. There's Andres Chang asked a question. Excellent talk. Your data on global HED scores is quite intriguing. I was wondering if your data set, you have information that would identify how multiracial individuals on any given continent and whether you're powered enough to tease out these things. So we're up to about 14,000 patients. This is a global survey that we're working on. And it's quite interesting. What we're doing right now is really looking at where diversity comes from. And there are certain alleles which drive diversity, which have been introduced into populations, right? So O201, it turns out to be a big driver of diversity in Western populations, but in Asia, it's different. And many of those populations, it turns out, this is unpublished data, but I'll just go ahead and tell you this. In Asia, a big generation of diversity. There's only about a half dozen really common alleles. So there's a founder effect compared to the farther you move away from Africa. And what we basically found is that they're linked actually to archaeo HLAs that we've inherited from different subspecies of humans, actually, different Neanderthal and Denisovian HLAs. And so there are a variety of, a few papers, high-profile papers showing admixture in different populations. And it turns out that some of those alleles are major drivers of diversity in populations in which there are relatively few alleles, right? So it's sort of like making up for the lack for diversity generated by pure numbers of alleles in any population. So really kind of cool. All right, another question here that popped up. Why in your graph does TMB not correlate with response to ICD and GBM? That's because probably in GBM, the drug just doesn't work at all. I've not had a single responder in GBM. So I don't think any biomarker will be useful in a situation where the drug just doesn't work. Ways out, how to improve ICI in immune cold tumors. It depends why the tumor is immune cold. A lot of times in pancreatic cancer, what we basically see is that there are certain chemokines and cytokines like CXCL4 and 12, which actually cause immune cells not to go in. I think there are a lot of effort being put on this now. Inhibiting the right checkpoints, inhibiting the right cytokine milieu is going to be very important. It's going to be much more difficult to overcome resistance due to lack of tumor rejection potential, right? There's no genetic basis for rejection. That's much harder. And there are studies now looking at how we can combine epigenetic therapies to reactivate peptides from ERVs and so forth. There's quite a bit of evidence now that ERV is involved in RCC exceptional response to immune checkpoint blockade. So those are some of the things that people are looking at. Where are we in terms of time, Pritesh?
I think we've got one more question, and after that, I think we'll be pretty good with time. So Dr. Waller asked, does the relatively poor correlation between HED and TAA diversity compared with viral proteins reflect a deletion of incipient tumors by the immune system before they become clinically evident and are accessible for sequencing of the tumor mutational load?
That's a great question. I think that may be. An alternative hypothesis is simply that our HLAs are evolutionarily driven and designed to present things that are very foreign, right? I mean, there are certain motifs in viral peptides that are very, how should I say this, very apt to be bound by the anchor residues of HLA, and our HLAs have hence evolved to bind those peptides because those are the survivors. They're not really designed to bind neopeptides. And so we've done some machine learning on this. If you actually look at physiochemical space and you do some machine learning, you see that neopeptides are sort of in diversity space halfway, a little less than halfway between clusters defined by viral or bacterial peptides and self-peptides. And so biophysically, they're just different. It's a whole class, class-wise, it's just different. And often they lack those motifs, those signatures in viral peptides that the MHC binding cleft, exon 2 and 3 binding cleft have really evolved to bind. That's my current guess based on the modeling. But it could be something else, I'm not sure, but very, very important to answer.
Looks like we have maybe one last question here in the chat that was posted earlier.
Oh, yes.
If HED is so important to predict ICB therapy, what does this mean for preclinical models? Example, mice in assessing novel therapies?
Yeah, preclinical models are not great to assess immunotherapies. There's lack of diversity. You see what happened now with microbiome, right? Like, the microbiomes are different, completely missed by mouse models. In fact, if you even look at the original mouse models in the data from some of Jim Allison's papers and so forth, it was not impressive. CTLA-4 was, compared to something like erlotinib, right? I mean, that's why it was ignored for such a long time. I do think that mouse models have a role because you can do controls, right? And you can isolate variables, you can test something. But in terms of how this translates into humans, I really don't think that we should really depend on them to make absolute decisions. There have been many cases in which something that doesn't really look good in mice actually is very good in humans, such as anti-CTLA-4. And there's many, many cases in which it looks great in mice and then just terrible in humans, like all the other checkpoints, right? So that we've tested, right? And so, I think, I don't want to really sort of criticize mouse modeling because we do mouse modeling, it's very important, but I think you have to interpret the data in a certain way, and you can't assume that humans and mouse models are the same. Humans are a very diverse set of populations all across the world. I've just showed you all this data. We have an immunologic history. We have genetic diversity, both at the level of HLA and peptides and ethnicity. And I haven't even begun to show you the data that's just beginning to be assembled on TCR diversity, right? The other third component of that trimeric complex. All these things are very homogeneous in mice. And so you're only capturing part of the story. It's getting better now because we have now humanized mice at least. And various people are trying to make more diverse mice with human HLAs and so forth. Jackson Labs even has some of these right now. So it's getting better, and the field is improving in terms of generating these reagents. But this is just me. Okay, I've always taken the mouse data for immunotherapy with a grain of salt.
Okay, well, Dr. Chan, thanks again, really for a wonderful talk, and thank you for offering to spend some time with our residents today as well. I just want to remind everybody, next week's Grand Round presentation will be by Dr. Corinne Faber-Finn from the University of Manchester. She'll be presenting on the modern management of small cell lung cancer, and you can see the entire calendar for Grand Rounds on the Winship website. Thanks again, Dr. Chan, and thanks everyone for joining.
Thank you so much. Bye-bye.